Alright, so now we need to wrap up central dogma with translation. Alright, so some key aspects of translation. Alright, so if we look at it, we've gone from DNA to our mRNA, right, and now we need to convert this to our protein. Okay. So we need to look at ribosome structure. Alright, so we need ribosome structure. We need to look at the actual code itself. Alright. And so we have the codon, right, which is part of our mRNA. And we have our anticodon that is part of our tRNA. Okay? And we need to also finally pay attention to the direction. Okay? And so the ribosome reads, right? So we're going to read the mRNA now. 5' to 3'. Okay, so those are just kind of some basic things that we need to iron out before we get too far. Okay. Let's start with the code. Alright, so the code is triplet. Alright. And there we go. So the code is triplet. It is, there are 64 possibilities, all right? And basically the way to think of it is that something like AUG, right, which is our start codon, right, that corresponds to the amino acid methionine. Okay, something that is AAA, right, that corresponds to lysine. Okay, and you should be able to look these up based on that chart that I provided you in class. All right, how to transfer in between. So the code is triplet, and so what we're going to have then is if we have our mRNA molecule, the way to think of it is that we've got, you know, these nucleotides, right, and some of them are smaller and some of them are bigger. Okay, and we're going to read it in threes. Okay, and that's how we're going to read it. Okay, now the other way to look at the code, all right, is in our tRNA molecule, all right, and so our tRNA, right, it tends to be this RNA molecule that has, let's not, I don't want to do that, all right, it tends to have stems and loops. Okay, and it comes up like so. Now, at the bottom, all right, so at the bottom here, there will be 3 bases, and we call that our anticodon. All right, and up here at the top of our tRNA molecule, all right, we add our amino acid. Okay, so this is a charged molecule, you know, this is a charged tRNA, so we call this thing an amino acyl tRNA, which just means that it has this amino acid attached to it here. Okay, and so and we refer to these as charged. Okay. Now, what's going to happen in the ribosome is that these tRNAs are going to be matched up with the proper codon. All right, and so the anticodon is complementary to the codon, and so we will develop hydrogen bonds then, right? Like, which would be something like so. Okay, so that's how we're going to match this up, all right? And so that's the role of the tRNA, is to carry these amino acids to the ribosome, all right? So the other thing that we really want to spend a little bit of time talking about is how we charge these tRNAs, right? How we're actually going to make this thing, all right? And so what's going to happen is we are going to use enzymes that are called aminoacyl all right, tRNA synthetases. Okay. Now, there is a synthetase for each amino acid, all right, and each tRNA. And so really all it is it's an enzyme, and so your book kind of gives you an example of an enzyme that kind of looks like an ear, all right, and so it has a hook shape, and then there's a little spot up here, okay, and so this is my enzyme, okay. What's going to happen is that my amino acid, okay, and I'm just going to make it a square to make it easy for me, all right, my amino acid and GTP, okay, sorry, we're not ready for GTP yet, sorry, it's ATP, all right, and I'm just going to make my ATP a little star, okay, and so what's going to happen is this amino acid, right, that fits into the active site is going to enter, our ATP is going to enter, right, and we're going to end up with then a phosphorylated amino acid inside of our enzyme, right, so I'm going to draw it again. Hopefully it looks somewhat like the last one, okay, and so why don't we change the color of our amino acid just to make it work. So here's my amino acid, all right, and to this thing now we will have attached a phosphate group, all right, so it has been energized, right, so that's really what we're doing, okay. Now what's going to happen next is, so we need our enzyme back again, okay. What is going to happen is we have our amino acid, okay, so it's still here, all right. Now, in this step, we're going to see a tRNA molecule come in, all right, and so my tRNA, I'm just going to draw it kind of like your book does. I'm just going to make it with a little bit L-shaped, okay, and it's going to come on in here, right, it's going to slide into place, okay, and as that happens, all right, the phosphate group that was here, all right, is going to leave, all right, so let's get rid of the phosphate group. Okay, and let's get rid of our little arrow, all right, and now we're going to put this guy in here. So we're going to put, and I'm going to orient it, it's more like this. All right, so here's my tRNA, and in between these two now we have a bond, right? So we'll put a bond right here, okay? And this no longer is here, right? Because it's now inside my active site. And once I have made this charged tRNA, my aminoacyl tRNA, it is now going to exit. Oops. Right? And so what I'll have then is my charged tRNA. Okay, and so there's my aminoacyl tRNA that has been made by my aminoacyl tRNA synthetase enzyme. Okay, and so now this molecule is ready to travel to the ribosome and take part in translation. Okay, so the next thing we need to address then is the ribosome, right? We need to look at its structure, all right? Your book seems to think the ribosome is brown, so we'll draw it in brown, all right? It has sort of this thing down here at the bottom, all right? And this is my small subunit. Okay, as part of the small subunit is a RNA, mRNA binding site. Okay, so I'll put that in here. Okay, that's my mRNA binding site. Okay. Then we have the large subunit. Right, and the large subunit kind of like so. Okay. And within this, we have 3 chambers, all right, or 3 sites or locations. All right, and I'm just going to draw these sort of like garage doors or something. Okay, and they are E, P, and A. Okay, A is the acceptor site, and so basically you want to think of your tRNAs, all right, plus the amino acid, right? They are going to come in to our A site, okay? Then in our peptidyl site, there is also a tunnel that allows our peptide to basically move out, all right? And so within the peptidyl site, we are going to have our peptide chain being held. Okay, so we've got the peptide growing out of there. And then we have the E site, okay, and which is where we are going to have our uncharged tRNAs exit. Okay, so those are the basics. The other thing that I have left off of this, though, is my actual mRNA molecule. Okay, so my mRNA has to come down here to my binding site, and it's going to kind of be passing through here, right? And we want to think of it as having, right, these nucleotides. Notice I've got these in 3s, so we're doing this by codon. All right, this is my 5' end, this is my 3' end. Okay, so that's the basics of our ribosome. Now, what we want to do is we want to look at how we would initiate or run translation. So translation has 3 steps. All right, first is initiation. Second is elongation, and third is termination, just like RNA synthesis, all right? Okay, just like transcription, so this is for translation. So we're going to start with initiation, all right? So the way initiation works is we're going to take our small subunit here, okay, so there's my small subunit. Right here is my mRNA binding site. Okay, so what's going to happen is our RNA molecule is going to come along, our mRNA, and it's going to bind in here. Okay, and so this is my 3' end. This is my 5', but I want you to think of it as running off the screen, right? Because it's longer, okay? Whoops, and actually no, it shouldn't run off the end on this end. Okay, we want to think of it as running off the screen on this end. Okay, Now, this whole thing is going to move in this direction eventually. All right. Now, in addition to this small subunit, all right, so what's going to happen then is that this mRNA molecule has now been positioned correctly on the small subunit. All right. We have our codons here, all right, and so I'm going to put just a couple here, all right. Okay, so here's this one, and this one's going to be our start. Now, what's going to happen then to our start codon is that we are going to get our charged tRNA molecule. It's going to come in, okay, and to this it has attached the amino acid methionine. So I'm just going to give it a circle, okay, and of course it has the complementary peptides here, and so we're going to bond these together. So we're going to have some hydrogen bonds here, okay, so we'll just kind of draw those in. All right, now once we have this thing oriented, right, and the methionine is in place, all right, now what we're going to do is we are going to have the large subunit is going to dock onto this thing, and so in comes our large subunit, like so, okay, and so now if I think about this, right, I have my E site, okay, I have my P site, and I have my A site, okay, and coming out through here is this tunnel where we're going to see our protein be able to be held, okay. Now that is initiation, okay, and so what we have is we have our first tRNA that is holding our first amino acid in the P site, in the peptidyl site, all right, and now we're ready to move to elongation, okay. So what's going to happen then is the next tRNA is going to come in, all right, and so it's going to be coming in from over here, all right, and let's just put it right here, okay, and to it is attached the amino acid that corresponds, right, to this tRNA, and we've got our anticodon here and our codon here, all right, and so now the ribosome is going to catalyze the synthesis of a peptide bond between the two amino acids, and this bond here is going to disappear, along with obviously part of my tRNA. So let's get that back. Okay, so now we have created a bond between these two amino acids, and now we need to shift this whole thing, right? We need to do what is called translocate it, right? So we need to translocate the ribosome, and we're going to shift basically one codon. So the codon that is now in the A site is going to be in the P site. The codon that is in the P site is going to be in the E site, right? So this one will move here, this one will move here, and this one will just be back behind us, okay? All right, so let's look at how this looks after we've done translocation, all right? So we need our, oops, wrong color. Okay, so we need our ribosome back. So here's my small subunit. Kind of looks like a hamburger bun. All right, and we have our large subunit. Okay, I also think this thing kind of looks like a snail. But it's a ribosome. Okay, so now we've got our E, P, and A sites, right? Okay. We need our mRNA molecule, right, running through here. Okay. And of course we need our codons. All right. Now we just translocated, all right, and so what we should have is a tRNA with the anticodon to our codon, and it should be attached, right, by a peptide bond, or not peptide bond, by a bond, a covalent bond to our second amino acid, which is peptide bonded to our first, okay? Okay? And of course these things are heading out our peptide tunnel here, right? They're working their way out this thing. It's out through here. Now, we're doing elongation, so then the next step is we need another tRNA, right, to come into the A site. Okay, and we'll add a different amino acid this time. Okay, the ribosome is going to catalyze the bond between this amino acid here and this one over here. Okay, this bond no longer exists, and we translocate again. And this continues and continues and continues on until we get to a stop codon. All right. So, one last time to draw up this thing. So by now you should be getting pretty good at this. You should be thinking, okay, he's drawing the small subunit, he's drawing the large subunit, he's drawing the exit site, the peptidal site, and the acceptor site. Okay? Okay, what should be in my P site should be a tRNA, right? That is holding on to a series of amino acids that is my peptide. Right, and I'm just doing them kind of quick and dirty this time. All right, so that's my growing peptide chain. All right, our mRNA is still through here. Okay, and we've got our codons. Now the key is this time, this codon right here is a stop, which means there are no tRNAs that correspond to it. All right, so question becomes, well, what is going to happen, right? And so what's going to happen is that the ribosome is going to pause, all right? And when it pauses, all right, that will allow for a release factor, all right, a protein called a release factor, all right, to come in, all right, and it will dock right in here. All right, and what it is going to do is it is going to break this bond here that is linking or holding our peptide in place. All right, that will allow then for our peptide to leave. Our large subunit will disassociate from our small subunit, and our mRNA will be free. Okay. This process requires GTP, right? This is going to require some GTP to get this done. And actually all this movement of the ribosome, the creation of these bonds, it all requires energy. All right? But the basic idea is we now have generated our peptide, okay? And so in our example, then, our peptide was just sort of these strings of amino acids, okay? And as you know, this thing has to be folded, right? It might have to be activated, which means it might need a phosphorylation, so it might have to have phosphate added to it, or it might even need to be cut, right? You might have to actually break a piece of it off to make it into its active form. So there's still some processing to occur, but that's the basics of translation.